Hello, this is Wildcat Science with Mrs. Haugo. This is biology. We are in cell biology, and today we'll be doing our cell biology notes. So please title your page Cell Biology Notes, and then title it Activity Number 13, the Cell Cycle. Okay, then please write down the challenge. What happens during each phase of the cell cycle, and how are these phases regulated? So please write that down and we'll talk about it. So we will together in class read page 229, and we should be able to figure out why cells divide. You don't have to write this, but make sure you're paying attention. So now please write this. Cells divide so you don't need to write the purple stuff, but please write the black. The organism can grow. Single-celled organisms can reproduce. Bodies can repair themselves. So that is why cells grow. So it's pretty important. So please write those down. So each cell has a cycle. So let's talk about that. The process of cells growing and dividing is called the cell cycle. And it is divided into two main parts. The cell cycle is divided into two main parts: interphase and then mitosis. The cell cycle needs to be controlled because uncontrolled cell growth and uncontrolled cell division leads to cancer. After each step, there is a checkpoint to ensure that everything's okay so that the cell cycle is under control. So let's talk about the first part of the cell cycle, interphase. During interphase, the cell grows and DNA is copied as the cell prepares for mitosis. The cell spends most of their lives in interphase. It is the longest part of the cell cycle. An interphase is divided into three phases: G1, G1 checkpoint, S, and G2. So let's talk about those. G1, the cell grows bigger. G1 checkpoint is where the cell checks to see if the DNA is damaged. So there's a check there. S stands for synthesis. This is when the DNA replicates itself. So now you have two copies of DNA. And then G2, the cell grows more and it prepares for cell division. There's one more checkpoint, the G2 checkpoint, and that is where the cell checks to make sure the DNA has replicated itself and it's done it properly. So let's talk about mitosis. So mitosis is the other part of the cell cycle. And during this part, the nucleus divides and results in two identical copies of the original cell. So you end up with two copies. They're identical. And mitosis is also divided into phases. There's prophase, metaphase, a checkpoint, anaphase, telophase, and then cytokinesis. So prophase, metaphase, we got to check that the chromosomes are aligned during metaphase. Then there's anaphase and telophase, and then cytokinesis. So let's look at a picture of the whole cell cycle. So you can see all of this yellow and orange stuff is interphase. It is the longest part. There's the G1 growing, the S where DNA is replicated, the G2 more growing, and then all the blue part is mitosis. We have prophase, metaphase, anaphase, telophase, and then at the end of telophase is cytokinesis, where that cell will split into two. Okay, please make sure you're writing down all the black stuff. Some cells will divide more often than others. For example, skin cells, a skin cell goes through that cell cycle about every 2, 3, or 4 weeks. Whereas a nerve cell never replicates. So you have to make do with the nerve cells that you have. So take care of your brain. All right, so then let's take a look at this other picture of the cell cycle. So we have G1, S, G2, that's interphase, and then mitosis and cytokinesis, and then Sometimes the cell isn't going to replicate, so it just kind of hangs out in this G0 or G0 phase. And then if it does, like a liver cell doesn't replicate all that often, so then it would enter it occasionally. All right, for activity number 13, make sure we're going to be doing the vocab, and we're also going to be reading the cancer article and doing post-it notes and doing the disease information worksheet about it. And then we're going to have somebody answer the challenge, which was what happens during each phase of the cell cycle and how are these phases regulated. Okay, we're going to talk about stem cell differentiation. Now you don't have to write this down, but make sure you're paying attention. So you are a complicated multicellular organism with many different types of cells. But you started out as one single cell, about the size of a period on a page. How did that happen? Well, you started out as a one-celled zygote, which was a fertilized egg, which underwent mitosis to make 2 cells, that underwent mitosis to make 4 cells, which underwent mitosis to make 8 cells, which underwent mitosis to make 16 cells, which underwent mitosis to make 32 cells, which underwent mitosis to make 64 cells, which underwent mitosis to make 128 cells. So please now write this down. Activity number 14, stem cell differentiation. So how do you get all of your different cells? Please write down the challenge. How do stem cells produce specialized cells? And now when we're writing this, you should either skip 3 lines or start a new page for a new activity. Activity number 14, stem cell differentiation. Challenge, how do stem cells produce specialized cells? So either start a new page or skip 3 lines from your activity number 13 notes. So in class, we're going to read page 236 to 237, and please write this down. Most cells are specialized, or this word differentiated, which mean the same thing. So differentiated mean it is a specialized cell. So a heart cell is a heart cell and can only be a heart cell. It is very different from a skin cell or a lung cell or an intestinal cell or any of the other 220 cells in your body. All of the specialized cells came from stem cells, which are a cell that has the potential to become any other kind of cell. So a stem cell has potential to do anything. So please write down the steps. The stem cell first undergoes mitosis to make another cell. So now there's a stem cell and a second cell. We call this second cell the daughter cell. It has parts of the DNA are either activated or suppressed, turned on or turned off. And so when certain parts of the DNA are turned on and other parts are turned off, we call this specialized. Something you need to know. After a cell has been differentiated, it cannot turn into any other type of cell. For example, a nerve cell, once it has differentiated into a nerve cell, can only be a nerve cell. It cannot be a heart cell. Only heart cells can be heart cells. So please write that down. And make sure you do, we will be doing our activity number 14 vocab. And we'll have somebody answer the question, how do stem cells produce specialized cells? Okay, so that concluded activity number 14. So please either skip 3 lines or start a new page for activity number 15, stem cell research. Please write down the challenge. What are the current scientific understandings and social debates about stem cell research? We will be reading page 240 in class. Please write down, stem cells can be totipotent, form any kind of cell. You have the potential to be anything. Or pluripotent, which means you can form most cells. Or multipotent, which means you can form a couple of different types of cells. And these are stem cells. Remember, once it's a specialized cell, it can only be one thing. So an embryo has totipotent and pluripotent stem cells. So an embryo has some cells at some stages of the embryo. The stem cells can form any other type of cell. And then at some stages of the embryo, the pluripotent, like it turns off some of its potential and it can only form some of them. So adults, kids, and fetuses only have multipotent stem cells, where a stem cell can form a couple of different types of cells. So by the time it's a fetus, a stem cell cannot do everything. It can only form, like, maybe it's a bone marrow cell, and it could be a red blood cell, a white blood cell, or a platelet. So what do we use stem cells for? Stem cells are used for research, and we have used them for researching and learning about diseases like Parkinson's, cancer, diabetes, heart disease, spinal cord injuries, and so on. So using stem cells, we have made a lot of progress on these diseases. Embryonic stem cells usually come from fertility clinics and are donated. So a fertility clinic is, so if a couple can't have a baby, they will like have a surgery. The woman will have her eggs taken out, and then the sperm, like in a lab, it'll be fertilized, and then they'll give the woman hormones, and then they'll actually like inject into the uterus or by the cervix the fertilized egg. And so then that is in vitro fertilization, and that's what those fertility clinics do. So let's talk about some misconceptions. So a common misconception is that the embryonic stem cells come from selling aborted fetuses. Well, there's a couple of things that are wrong with this misconception. As we know, fetuses do not have totipotent stem cells. They only have multipotent stem cells, so their cells are too specialized to be good for research. And also, you can't sell fetuses. The stem cells can come from donating. Like if you have had your children and you're done with your embryos, then you can either donate them or have them destroyed. So again, women have the choice to donate fetal tissue for research. You can't sell it. And that has been going on for a while, since the 30s. And some things that have come out of this are vaccines for polio, measles, mumps, rubella, hepatitis A, hepatitis B, shingles, rabies. All of these vaccines have come from research on fetal tissue. Okay, so we're going to talk about the difference between an ethical and a scientific question. So an ethical question is one that science can't answer. It's like a right or wrong question. A scientific question is something that we can figure out doing scientific research or scientific experiments. So what I want you to do is you don't need to write this statement down. You just need to write down ethical or scientific. So number 1, ethical or scientific. Do clones ever occur naturally? Number 2, should our government invest money in stem cell research to help cure diseases? Please write ethical or scientific. Number 3, how can stem cells be used to treat Parkinson's disease? Ethical or scientific? Number 4, what are the potential drawbacks of using embryonic stem cells for research? Ethical or scientific? Number 5, is it okay to use an embryo for stem cell research if it destroys the embryo? Scientific or ethical? Number 6, how does therapeutic cloning work? Scientific or ethical? Should patients choose the traits of their future child if we can clone humans? Ethical, scientific? Number 8, can a woman be paid to donate her eggs for a stem cell research? Ethical, scientific? What other diseases could be treated using embryonic stem cells? Ethical, scientific? Number 10, what rights would a cloned human have? Right, ethical or scientific? We will go over the answers in class and correct them. So to finish up the unit, we'll do vocab and our challenge. All right, good job on your notes.